Okay, yeah, so thank you so much for joining and good evening, everyone. It gives me a great honor in having Yasin Jeshima. She's a senior scientist at ICAR Pusa. She obtained her PhD and postdoctoral years of experience later working for Israel and several other places, working in the areas of crop improvement and genetics. If I look into her illustrious career, she has had profound and prolific scientific exposure. I'll probably read out very few important things so that she has gone through her period of research career. Apart from her illustrious PhD and postdoctoral years of career, Yasin has been invited in at least as many as 100 plus conferences, and she has won as many as at least 40 plus best presentation awards. Apart from at least 4 or 5 gold medals that I could see here. This is an absolutely prolific scientific galore that I can talk about Yasin, and I've known her over the last 11 years at least, of course through Bioclues and later on through the various conference series. She has been very actively participating in all the events, and she has been an amazing speaker, amazing I would say orator, and of course scientist par excellence. And she was in fact deputed to participate in consultative meeting for an expert group on synthetic biology way back in 2017, and for which since she is an NBPGR, she is not only responsible for maintaining the digital sequence data pertaining to genetic resources through NBPGR, but also taking course of synthetic biology entities as well. And she has at least executed more than 10 crores of grants, and this particular introduction needs no more accolades, but what we usually do in Sci'versation is we request the guest speaker to speak for the first 10 minutes on how their particular career unfolded, and then there will be questions in extempore from me. Okay, so with this little introduction, I thank Yasin Jeshima for making her time today, and over to you, Yasin. Please share your presentation. Thank you.
Thank you, sir. Thank you so much for the very elaborate and very powerful introduction. I have to match your introduction. That much I have to show today. It's my pleasure to be here, and I'm highly excited to be here. Before that, I just like to take a moment to introduce myself before getting into the scientific achievements or scientific work what we have done so far. So, shall I share the screen?
Yeah, please. Yeah, please.
Okay. I'm sharing my screen. I did like to take a moment to introduce myself. It is not just through the academic titles, but through the journey and the people who made it meaningful. I was born and brought up in a Jamindar's palace near Madurai. I'm a great granddaughter of a Jamin. And my early education was rooted in the values of that upbringing. Began in the schools of Teresa Convent at Madurai. It is near Teppakulam region of Madurai. Those formative years taught me discipline, resilience, and focus. Like it was very stringent system of schooling. I pursued both my UG as well as PG degrees from TNAU. That was supported by State Merit Scholarship. Though I got qualified for JNU JRF in biotechnology, I chose to stay with TNAU and later joined Kerala Agricultural University for my PhD with ICR SRF. During my third year of my PhD, I returned back to TNU as a senior research fellow. I joined at Curriculum campus. That was my final year of my doctoral journey, and I was fortunate to be selected for multiple prestigious positions during that time. I got selected for ARS, TNPSC Group 1, AO in TNPSC, and Assistant Professor at Kerala Agricultural University. I was a little bit confused to choose which one, but at last I chose ARS and began my career at ICR in literature. Till then, there is no gap. Meanwhile, after my PG, till the time I took PhD, some gap was there, somewhere around 6 7 months was there. At that time, also, I was working as SRF in GNU. During both these time, like before PhD and during last year of my PhD, I was working in rice. First, I was working in a hybrid rice program of TNA Venture Capital, and then during my third year of my PhD, and after immediately after completion, thesis submission and all, I was working in salinity tolerance of rice. So that was my initial scientific research work project. It means a Pakka project. Okay, so initially 2 projects in GNU, and then 2 projects when I was in Kilikulam. And one was in BRNS, another one was from DST JSPS. It was Indo-Japan project. So very beginning itself was my international collaboration. So starting itself international. So everything is international here. Then after coming to NDPGR, it's more than a decade now. So it's all like means no going back. Okay, so I'm not looking back to why I have chosen this or anything like that. Nothing like that. So after coming to NDPGR, during my in-service, I got postdoctoral fellowship. Actually, I got 2 opportunities, one to US and another one is to Israel, but I chose Israel. I thought like that will be a power-packed one. But it was, the experience was a little bit different, but what I expected is more, but it was like what we talk about that country is a little bit exaggerated one. What we see there is a little bit less only. Okay, that is not that much a satisfying one. But I learned a lot of things. I learned a lot of things like there is no need to have a lot of elaborate things, means lot of things in the lab to have a very good publication. That is the first and foremost thing I learned. So all those people who are publishing in Nature and Science, they had only one PCR and one electrophoresis in their labs, and everything else is all in centralized facility. That is the first lesson I learned. So don't try to keep a lot of things in the lab. First thing I learned is that one. And okay, there are so many things. Apart from that, I studied in Weizmann Institute. That is a very, very big turning point in my life. Means the things what I learned there has changed my way of seeing things, way of doing research, as well as way of seeing things, and how I react. Everything changed after learning from Weizmann Institute. Later, I got many opportunities to go abroad. I had one Indo-Swiss collaborative project that was great support. That is my first individual externally funded project as PI in ICR system. And after that, I had opportunities to go to US, UK, and Switzerland. It's all through externally funded projects only. So now we can see like means these slides are up to 2020. After that, I have not updated this slides. If you want to go through, then you can just go through my ResearchGate as well as Google Scholar profile. These are all the things I just wanted to highlight. That's why I'm just maintaining that one. This one actually is a best example, like how we develop new markers and how we go about it. Okay. And apart from developing new molecular markers, we develop genetic stocks also. This is one such example. This is somewhere around 60 to 75 centimeter tall. And at that stage itself, it started flowering. But this is under stress condition. And we develop browser tracks like this. And then apart from that, we work on breeding as well as developing other molecular biology tools as well. Okay, so these are all some of the examples. And I'm known for these kind of diagrams. Most of my papers will be having gig networks. So I work in system biology approach. So for that example, I have given here. Not just directly on the plants, the impact of pest and diseases as well as stress on plants. That's what we look for. So we have identified many genes and co-expression, differential expression, gene network. These are all the kind of things I work. Uh, okay. So apart from that, we have done some work in black rum also. I told rice, but I have not seen shown here any rice. Uh, One such example is we are the people first who developed genome edited rice lines, but we were not successful to bring that into a commercial line just because it was coinciding with the Corona period. So I did this work. Sorry, actually, I collaborated this work with my mentor, Dr. Pillai sir. So he is actual Brahma of that genome-edited rice. So far, I have published around 70 papers, presented more than 100 conferences, means 100 conferences presentation. I have written around 8 books and contributed more than 10 book chapters. I don't know exactly the number because I don't keep a strong record of my CV. So that I have to do. There I'm lacking. I have received more than 30 awards, maybe 32, I think. And so far we have completed 6 externally funded projects and 4 in-house projects we have completed, and four now it is running. 4 or 3, I think 3 it is running. So I already told you that my base has been in PUSA for more than a decade, and here I teach PG students also. Here I teach bioinformatics as well as statistical genetics. Yeah, this is the one means the genes what we have validated by genome editing. So we used the genome editing tool for validation of this one. Then some maps we have developed, something we have identified. This is an example of a database what we have developed, and so many other markers what we have developed. Though my work keeps me fully engaged, I'm most touched by my another role, that is delivering motivational lectures. I have been invited to speak by many institutions, and what truly humbles me is hearing from students who are being inspired to push forward and succeed. So if they believe me, uh, I also believe them. I believe them more than what they do. So I motivate them. I make them successful by following my words. Okay. So I think um, I don't know how to express thanks to those people. Like they follow my words. They take each and every hint of me. They follow my words. They do it. They excel in their field. They come up with a bright future, and they say they all of a sudden they call me and say like, oh, I cleared this exam. I got this post. I'm going there. So these kind of words I have heard from means since 2017. I am hearing these kind of words from many students. I think somewhere around 500 plus successful students I could see. So 1 hour lecture, if 1 hour lecture can make a big change like this, I think I can do a little bit more to the society. So that's what I believe in myself. So as you know, I'm not a social media person. I'm not present in any social media. You can reach me only through my professional pages like ResearchGate, my institute profile, or directly emailing me. So that's what we can do. So my time and energy are wholly devoted to science, teaching, and mentoring. So while I speak about my journey, I must also speak the people behind this journey. If you consider my life a successful one, that success belongs to my teachers and professors. So I would like to express my deepest gratitude to Dr. Vivekanandan, who was my advisory committee during my master's, my post-graduation, and later he was dean when I was working as SRF. That is means during my time as SRF. Then Dr. Maheshwaran, he was former director of research in TNU. He was my UG teacher. The subject knowledge, inspiration, what I carry today stem from him. Sir continues to be my guiding force. And the most important is Dr. Arumugam Pillai. He was my mentor. If I faced life with a calm mind and a peaceful smile, it's just because of him, his influence and his training. So I trust in people. Uh, I trust everyone. I think almost everyone. That is just because Dr. Pillai. Um, Before meeting my mentor, Pillai sir, I never trusted anyone. I don't trust anybody. But after meeting him, the way of seeing the world has changed. So that is the impact of a teacher. That is the impact of a professor. And now, very specifically, I have to mention Dr. Vishwanathan, Joint Director of IRI, who has been a source of guidance and mentorship since my college days and continues to be a steady influence on my path. This list won't be complete. It's a very long list. But I have to mention one school teacher, Dr. She's not doctorate. She's Mrs. Saroja Devi. She was my third standard teacher whose yearly lessons planted the seeds of who I have been today. Okay, who I have become today. Though I have named only few, my gratitude extends to every teacher and professor who has shaped my life. So the influence is means what you are seeing today is the impact of my teachers as well as my friends. Only that circle has made me what I am today. I carry their influence with me every day with pride and humility. I also take this opportunity to thank my directors and colleagues, past and present. So their continued support and encouragement made me to stand in front of you today. And their collaboration is a result of what you have seen in my profile. Their presence and trust have been instrumental in each and every achievement of me, whether it is a small one or a big one. This is all about brief of who am I, who I am. I hope today's session will bring some more impactful insights. Let us hand over to Dr. Prash again. Sir, over to you.
Thank you. Thank you so much, Yasin, for a wonderful introduction and a very modest and very humble, presentation that you have had. I mean, moving all the way from, Madurai in Tamil Nadu down south to Delhi. I mean, you have reached, pinnacle of success. That's always a kind of an inspiration. And your illustrious career, speaks volumes about the kind of, mentoring drive that you have had and how you translated that kind of knowledge that you have gained from mentoring and passing on the baton to the current generation. I mean, absolutely laudable, Yasin. Thank you so much for this. I have few questions. One of the major things that India is going through is smart agriculture. So if you really talk about the kind of the resilient programs or the sustainable development goals that we are going through, and yet we couldn't capture.
That term was coined by us.
Oh yeah, oh wow, wonderful, yeah.
Yeah.
So
In 2013, actually, we did that paper. Means it was in IRA hostel. So I was in Varsha hostel at that time. I myself with my friend Ramya and her husband. So all three of us were discussing. We were discussing something from Swaminathan sir's paper. Something we were discussing at that time. I was having an ordinary phone. I don't have smartphone at that time. My friend was saying everything is smart, not to your phone. She said like that. So I said like everything is smart, but not our agriculture also. That's why I'm not having any smartphone. So that's how we started using that term. But that time, same year, this UN has used that climate smart agriculture kind of thing. Climate smart term they used. They also published the paper or book, I think. It was in a booklet. Similar time, somewhere in November, after we have communicated that concept, they published it in November. our paper published in December.
Wow, wow.
We were the people to first to use smart agriculture and what is smart agriculture, how it is to be used. But later, many people wrote so many things on that. They spoiled the concept. It gone like somewhere misleading terms they used. But still, I think so. I have to write some more papers on smart agriculture. Then it will become strong.
Right. No, wonderful. I mean, if you really look back, that's a great thing that you are the one who has coined smart agriculture. But my point was something on those particular lines. Smart agriculture, resilient crops, or stress evasive crops, or something like those particular crops that can evade abiotic or biotic crops, or so called the smart
Tolerant,
crops. Yeah,
tolerant.
yeah, exactly. So they all go hand in hand. But India, with such a vast diversity, we can bring up a large amount of such resilience associated with crop improvement. So given the current scenario, where do we stand, Yasin? I mean, where do you think that India is with respect to development of such robust technologies, where we can.
Actually, to be frank, to be frankly speaking, we don't need any technologies. We do have enough resources. That's what I have to say. We do have lot of genetic resources. For example, if I take pigeon pea, we do have 16,000 plus successions in our gene bank. That is more than sufficient enough to answer all kind of stresses and other things available at present condition. So in future, like just Bible says or the Quran says, like in future there will be severe drought when the world is going to collapse at that time. So if we think future specific, then we can go for something, some improvements. But technology wise also we do have enough technologies, and resources wise also we do have enough resources. Only thing is we have to preserve it, we have to conserve it, we have to use it properly without contaminating it.
Right, right, wonderful. And moving forward, Northeast India has a large amount of orchids, which probably nowhere in the world we could have. I can see that Southeast Asia, including Indonesia and Malaysia, and of course Northeast India has got a large number of orchids. The other day I was just talking to one of my postdocs.
Western Ghats also is having lot of orchids.
Yeah, no, what I was saying is, if we can develop that
Still,
kind
it
of
is
a.
not even named, and it has replaced the place of Fabaceae. So Fabaceae was the second largest family, but it was replaced by orchids.
Right, right,
So that many orchids are there, and orchids are highly specific in their growing condition. So it cannot be brought into Delhi or somewhere very hot. So it needs very specific condition. It needs uh, they are not free living organisms. So the way we have to grow, it grows like a saprophyte kind of thing. So
right, right.
yeah, you can please come to
Yeah,
your
yeah,
question.
no,
I
no,
think I
no,
interrupted.
no, thank you, not at all. So what I was saying is, Yasin, I mean, since you work at NBPGR, National Bureau of Plant Genomic Resources, so do we have that, what do you call, tagged orchids, across the repositories? Do we have a digital, what do you call, profiles of all orchids that are available from, say,
For
India?
orchids, we do have a different institute. Separate institute is located in somewhere in Shimla, I think. I don't know exactly where it is located, but we do have one ICR institute specifically for orchids, and some digital libraries were made on our Indian orchids by foreign authors. I have seen their books. So in somewhere around 19 or 20, after 20, I think 2020, I have seen one new book on new catalog of orchids. They used our Indian people, local people to collect orchids. They labeled it, they named it, they took a photograph and they made it. So that kind of work our Indian scientists are also doing, but what exactly they are doing, I don't know. Just that belongs to their mandate.
Right.
So it is there, work is going on, conservation is happening, they are having means active germplasm site also. Means they do have a collection, so everything is happening.
Right, right, wonderful. And how do you maintain the germplasm, the robust germplasm at NBPGR, Yasin? What are the current challenges that ICAR is probably, looking into development of these?
No, no such challenges. Means they have answered all those things already. So we do have national active germplasm sites. We do have medium-term storage. We do have long-term storage, and we do have 10 regional stations, and we do have collaborators from active means active germplasm sites. So it is conserved in multiple locations in different forms. Like seed or the live plant is also in active germplasm site means it is the live plant, it is a field. So that kind of thing is there. And means every regional station is also having a medium term storage. So it's all there. Enough facilities are there, but maybe they may think about having one more duplicate of gene bank or additional facility like that.
Right, wonderful.
That is a policy issue that we cannot
Right,
say.
right, wonderful. Yeah, and moving forward, Yasin, moving to the real genomic science that that you have steadfastly worked on. I see you have worked on wonderful crops, several series of crops, and you have also deciphered the knowledge behind understanding the non-coding RNAs as well in some of the crops and the plants as well. So if you really look into the plants, they're largely duplicated. 30 to 40% of the genome is largely duplicated. You find a large number of repeat expansions, a large number of those chunks of those duplications sitting over there. Earlier, if you really see, when the next generation sequencing came into picture, people thought that okay, Illumina, could be suffice, and the short read sequencing or sequencing, methods that were employed using Illumina could have been, nicer. But over the years, the long read sequencing has taken, leaps and bounds, for the plant genomes. But one striking thing I always ask plant biologists is that we have never really looked into the plant exome capture. We only looked into the plant genome capture. And is there any particular reason why plant exomes are given a kind of a different dimension?
Not that much famous in India.
Yeah.
Even the hybrid approach is not applied everywhere. So when they are doing genome assembly, it is better to have a long read sequence as well as Illumina sequence as well, means Hanover as well as Illumina together, and go for hybrid assembly. So that one can address all these repeats region and kind of thing.
Right. I mean, I know about hybrid assembly just because plant genomes are
But that is not even done here. Means like only in one or two crops they have used it, and it is not popularly utilized yet. The reason behind that is financial issues.
right. Right. But
Maybe
yet,
funds could be the only reason behind that.
yeah, I mean, instead of recapitulating the genomic resources or redoing the whole genome sequencing of so many plants, I mean, taken from say short read to long read, and again sometimes high C. Many of those particular genomes were done with high C sequencing as well. I mean, that is a kind of a very Herculean efforts as well. And many species, subspecies are also carved out as well. And I'm sure there is a hope in the near future that maybe we can think of
Sequencing
machine learning. Yeah.
Sequencing kind of projects are not getting funded nowadays. That is a major lacuna in this.
Right.
So we need at least a few more projects. And one more thing is they concentrate on only few crops, but we are looking for some other crops.
Right.
So little bit of out of the box thinking is needed to be applied here.
Right.
Means funding agencies should give some relaxation in giving grants.
Right.
They should not narrow down only to rice or wheat or agency. They should come out with the open mind to give funds to many other crops also. So that is needed because our Indian crops are not important to everybody abroad. Why
Right.
they will work on our crops?
Right. Right. So moving further, Yasin. I mean, please tell us about how genome editing approaches in your lab or maybe in ICAR is going through. I could see a large amount of genome editing approaches are always on the anvil at ICAR. So can you please describe one or two shrewd examples that you have taken lead in these directions? Yeah.
I have done we have done genome editing in rice. So we used it to prove that means this particular gene is working in rice, uh, impacting in plant height and early maturity. So that we have already done. Uh, Means other labs, I don't know how they are working. So without knowing that how they are working, I cannot comment on that. But otherwise, this tool is important. We should have the technologies ready. When there is some crisis happens, then we should be ready to handle it. So once it is ready, then they can apply it. So not necessarily to release varieties, at least they should have a standardized technology. So this project is important. There is no doubt in that. But whether we should have all those varieties released immediately, we should think about it.
Right. Right. I mean, the other day I
We
was
do
just.
have diversity.
Yeah.
So I like to say that we should first use the available variations, germplasm resources first. We have to. So whenever PG students or PhD students are doing some research, it is better for them to first approach NBPGR to get some material from gene bank so that they can start with a very robust material or very useful material or very diverse material so that they can lead to any variety release through their university. So that
Right.
kind of approach is needed.
Right. I mean, the other day I was just talking to my good friend Velu. Of course, you were also there as a part of a workshop. We were talking about reverse what do you call breeding, and
Uh,
we were also talking about double haploids, and
yes.
of course now many of these particular monocots and the kind of germplasm that we have been making up in India, especially through Indian Council of Agricultural Research and Indian Agricultural Research Institutes. We have had sufficient number of those things that have been captured in germplasm. So, what are those efforts currently in developing new varieties through genome editing approaches? I mean, where are we placed in those particular directions?
Genome editing or double haploids.
Double haploids,
Double haploids we don't do.
right?
And in BPGR, we don't do any new breeding technique. So only up to pre-breeding is our mandate. So breeding is not our mandate. We are not supposed to release any varieties directly from BPGR.
I see, I see, yeah.
So that we cannot do.
Wonderful, yeah.
And double haploid is a very old technique. Like it is not. Nowadays we do have different methods to bring variations. So no need for double
Right.
haploids.
No, but even otherwise, I mean, in germplasm, I see, you always have these rils and nils that are maintained, across those, different crops, including the cash crops and all those kind of things. So, when you do reverse engineering or reverse, breeding approaches, some of these particular, varieties that are placed across those particular, heads, could be useful, for your research or maybe deciphering the genome editing approaches in the future. Yeah, so that was my point. Yeah, but yeah, but thank you, Yasin, on this. Final few questions. This is a very generic question that I usually ask every guest. So were there any disagreements that you put up in an agreeable way with anyone?
In what way?
Any scientific discussions that you think that you have disagreed and put those disagreements in an agreeable way, so the reason why I'm putting this is, mostly, for the current generation, where they can, emulate, from their mentors, how to agree to disagree. That is a very important, aspect, which I feel, with due respect to the current generation, they miss that. Yeah. Yeah. So if you can give any examples, on any scientific sojourn that you have gone through, any kind of an examples that you can narrate. Yeah.
I have not framed any such opinion like that.
Okay.
First thing is, I stopped being judgmental. I don't judge what others think or what others do. That is the first thing. Second thing, I concentrate mostly on my work. Then third thing is, for peace of mind, after 2020 Corona period, I stopped watching news channels. So on a overall way, I don't have anything to think about to disagree. So specifically, I don't have anything such like that.
Right. Right.
If
Wonderful.
you are having anything specific, means if I have to give opinion on anything specific, then you can ask me directly.
Right. Right. Yeah. So I also see a very important word called orphan legumes that probably your group has coined. Can you please
Yes.
tell us a little bit about that? Yeah.
So why we say orphan legumes means that it's all belongs to India. So we are the people who consumed it, we produced it, we exported it, we sold it, everything what only we did. Now, if we don't produce enough of those orphan legumes, I should not use that word orphan legumes. We should use underutilized legumes or underexplored legumes. We can say like that. So we have to concentrate on that. Like, but for the past few years, most of them are being imported. So that cost uh, high on our economy. So we have to concentrate on that. That is the major opinion about orphan legumes. Otherwise, like it's all very important. One thing I want to say is some compounds which is actually present in the dopamine pathway, like the things what it's what makes us happy. It is present in these orphan legumes. So either you have to means you have to make yourself happy. You have to feel yourself happy. Otherwise, you can eat non-veg. That is one way because some of the compounds are present in that pathway. Otherwise, take legumes. Take tur dal or horse gram. If you eat any of these things, you will be happy automatically because the compounds present in dopamine pathway is present here.
Right. Right. Wonderful. Yeah. So this was absolutely amazing having Sci'versation with you, Yasin. In fact, I've known you for almost, as I told you, 11 years, more than 11 years. It's always nice working with you, discussing science, and you have been the most active person in Bioclues that I've seen over the last 20 and a half years or 21 years. Thank you so much for this.
There
But
are questions in chat, it seems.
yes, yes, I'm coming to that. So,
Okay.
would anyone like to unmute your microphone and ask a question to Yasin? Yeah.
There are so many questions here, I could see.
Yeah. So, the very first question, Bopen, please unmute your microphone and you can ask. Yeah. Bopen Singh? Yeah. Yeah. Bopen, can you please unmute your microphone, please? Yeah. I can do that for you if you want. Yeah. Yeah. So he asked a question. Please let us know if you have any idea on the progress of the work on black rice in India.
Yeah, black rice is Nivara rice. Swaminathan Foundation has done a lot of work in black rice. Actually, this black rice belongs to some regions of Northeast as well as our Western Ghats. So Kerala is having a lot of black rice varieties as well as South Tamil Nadu is also having black rice. like kauni, and there are so many traditional varieties in rice. Not just black rice. There is one more rice we call matta rice. So actually, I am 50 percentage Mallus. My father is from Malayali background.
I see.
Yeah, Nambudiri's family, and my mom is from this Tamil Iyengar's origin. Actually, so both are freedom fighters families. So
I see.
actually, we our family itself has cultivated these black rices, and I have seen those black rices from my feet also.
Right.
So a lot of work has been done, and if anybody wants to have black rice, if it is a young person, if you need, you can take from NBPJ or you can take from Swaminathan Foundation.
Right. Wonderful. In fact, during my last 4 years
Tamil
of
Nadu
my
is having a field also. Like MS Swaminathan Foundation is having some research stations at Wayanadu, I think, and those stations they maintain in field also these black rices.
right.
So
Yeah.
if anybody wants, can contact me. I can help.
Wonderful. I mean, on a lighter note, during my last 4 years of my stay in Kerala, since I was growing very fat, someone advised me that you should have matta rice. So,
Yeah.
you'll become very thin. And of course, I don't take rice. I mean, I was just trying to convince that I was growing fat not just because of eating rice, but maybe because of lack of exercises, because of sitting on these chairs, for
With
almost.
matta rice, actually, if you eat matta rice, no mineral deficiency will come. No
Right,
deficiency, no calcium deficiency, no iron deficiency kind of thing.
right,
So, yeah.
right,
Only thing is it is boiled rice. That is
right.
the only.
Right. Yeah. So I think that answers your. I hope that answers your question, Bhupen Singh. And Pranita Batele asks a very important question. In fact, she's, of course, we're all working together on Bergam, of course. So Pranita, rest assured, Yasin is there to drive us for that Bergam project. So we're all working together on that. Yeah.
Yes, you can write to our director. There is one MTA available in our NB Teacher website. So once MTA is filled and signed, you can directly send to our director. They will arrange for the material. If there is any help you need, you may contact. Just I will go and check what is the status of it. I will find out.
Right. Wonderful. Wonderful. Yeah. So, assuming there are no questions, are there any quick questions? So, Yasin, I must thank you, for all the time and energy that you have put in today. Thank you
Thank you.
so much for your lovely introduction and gracious presence, and including your Prithvi Naga. Please, yeah, you can ask your question. Yeah, you can unmute. Yeah, yeah. Please unmute your microphone.
Uh,
Yeah,
Good evening,
Pavitra. Sorry,
sir.
Pavitra. Yeah, sorry. Yeah.
Hello.
Yeah, please go ahead. Yeah.
Good evening, sir. I'm a PG student.
Okay. What you need?
I'm Pavitra, ma'am.
Okay, it's okay.
I'm from Satyabama University, Chennai. Uh, Currently, I'm doing my
You come to
master's.
your question directly.
Okay, I'm really sorry. Ma'am, I'm doing a project based on biofortification.
Okay.
Regarding that, I have several doubts. I don't know how to approach you. That's why I'm asking how to approach you, ma'am, through mail or.
Just send me one mail. I'll give you my phone number.
Yeah, okay. My mail is Pavitra.
Wait. I'm giving you my mail ID.
Okay, ma'am. Okay. Sorry.
My mail ID is in. I'm just posting my mail ID.
Yeah. So as she types this, are
Yeah.
there any other
Those who
questions?
need help directly text me in the mail.
Yeah.
Okay. Thank you so much, ma'am.
Okay, fine.
So, friends, please switch on your camera, everyone. We'll have a very good photograph with Yasin to capture this, great moment. Yeah.
Sir, you are muted, not audible.
Oh, sorry. Sorry. What I was saying is, I was just requesting everyone to switch on their cameras so that we can have a lovely photograph. If everything goes on well, I take probably double pleasure in telling you all that there is Sir Richie Roberts in this particular group. He just left the group. He is a Nobel laureate for alternative splicing way back in 1993. He was listening to this, Sci'versation, and I thought of taking a moment to capture a photograph with him. So that's a great honor. We have at least 4 Nobel laureates in Bioclues and several Lasker awardees in Bioclues, and they all keep checking our emails. I hope we are not spamming them, but this has been a kind of the best Sci'versation with Yasin that I could think of in recent times. And friends, Yasin is always nice. I'll tell you one important thing about Yasin. We recently organized a workshop. Okay, she'll not let you sleep until she finishes, until you all finish her exercises.
As also I do the same thing.
Yes. And मैं डर के मारे में भाग गया था. I ran away because she would probably tell me that that I haven't done those exercises. Anyways, thank you so much, and everyone, please can you switch on your camera, please? There are only very few who have switched on your cameras. Please, yeah. Oh, good to see Inderjit Bogal after many days, yeah. Senthil, thank you so much, Senthil. I owe you a Sci'versation. I'll get back, yeah. Bopen, thank you, Banu sir, for joining. Thank you so much, yeah. Akshay, Yasvini, Kanvi, Pavitra, Guneshwar Bera, Usma, please, yeah. Praveen, yeah. Okay. When I call, people run away, so I'll just take a photograph. Okay. Yes. Thank you so much, and let's give a big round of applause to Yasin, please. Huh? Yeah.
Thank you, sir. Thank you so much. Thank you for the opportunity as well.
No, thank you. You're so down to earth, Yasin.
Thank you.
Thank you so much. Yeah. Thank you.
Thank you, Ananda.
Thank you.
Thank you, ma'am. Thank you, Dr. Prash, for this conversation.
Thank you.
Thank you.